a microphone. Okay, so the key thing here is that you're going to now target the specific tumor cell of interest. But the problem, of course, is you need to have an antigen. You need to know what the antigen is to use this. And because everyone's tumors are different, it may be hard to come up with an antibody that you can use in this case. People have also realized that we can, in fact, also just deliver antibodies against some of the antigens that are found on tumor cells as just regular old antibodies, and allow things like NK cell killing, complement-mediated lysis, things like that. So we don't always need to even have that payload conjugated onto the antibody. In fact, if we just deliver antibodies to the tumor antigens, that is often enough to eliminate the tumors. On the right-hand side, you can see some examples of antigens that are used in clinical settings for a number of different situations. One of them that I just want to mention really quickly, because it's going to come up as an example on later slides, is shown here, rituximab. So you can see one of the many antibodies that is in clinical use is rituximab. Rituximab is an antibody against CD20. And CD20 is on mature B cells. And so rituximab is used in cases where patients have certain types of leukemias and lymphomas where their tumor is a B cell. And so they have this B cell that's growing out of control. So we use an antibody against CD20 that's on that B cell to kill those tumor cells. Good news, we've narrowed this down and are not killing every single cell in the body like we might have killed with radiation or chemo. Bad news, yeah, we narrowed down, but we only got as narrow as now we're killing every B cell in the body. And so rituximab, while a revolutionary treatment, again, is going to be a bit immunosuppressive, and it's still not really narrow enough. And so what the other sort of general theme you'll see with a lot of these therapies is that we've done a great job going from really broad to way less broad, but we haven't gotten as narrow as we might need to in a lot of these cases. And so again, we've gone from suppress everything to suppress B cells, or kill everything to kill B cells. That's better, but you're still killing all B cells that have this on the surface. And so that's one place where people are really working to move forward in a lot of different therapeutics. If you recall from our discussion of cancer immunology last time, one of the particular problems that comes up if we're thinking about cancer and cancer therapy is that in general, when we are actually at a point of diagnosing a tumor, that tumor is evading the immune system. I talked a little bit about this idea last time, or at least I happened to mention the phrase. Sometimes people talk about certain tumors as cold tumors. And the basic idea of a cold tumor is that it's a tumor that can hide from the immune system. It's a tumor that's not stimulating an immune response. And so the idea is, your immune system can do things to fight tumors. In fact, we see that happening in those earlier phases, like the elimination phase. But eventually, we get to this point where the tumors figure out a way to evade immune responses. And so one other kind of general idea with a lot of these therapies is figuring out how to wake up the immune system, how to get that immune system that is getting evaded or that has turned off or that is cold and asleep back turned on. Because when it was turned on, it was actually useful. It's just that it got turned off. And so the other sort of general group of therapies that people talk about are therapies that are really basically trying to turn that immune response back on that has somehow been turned off previously. The most famous and successful of these therapies are described on this slide. These are, again, antibodies, and you'll see a lot of examples of antibodies today. If you recall, we talked about a situation where we have T cells needing signal 1 and signal 2, but we also can turn off our T cell by using this molecule CTLA4, so the T cell can get turned off. Sometimes what some researchers will do is they might use an antibody, like an antibody against CTLA4, and you're going to hear about 2 different CTLA4 antibodies at different times today, so pay attention to what the antibodies do, because they do different things in 2 different situations. Here, the CTLA4 antibody is what's known as a blocking antibody. So it's basically binding to CTLA4 and not actually making CTLA4 signal. It's getting in the way so other things can't interact with CTLA4. So it's sort of like sterically hindering CTLA4 from interacting with other stuff. You will see a slightly different antibody against CTLA4 later that actually tries to send a signal through CTLA4. This one's not signaling, it's just blocking. Alternatively, we have other negative regulators, and so the idea here is that the T cell never gets turned off, it stays on. So basically T cells that would otherwise have been off are now permanently on. So in the cancer situation, the problem is your T cells get turned off. And so if we block off signals, then perhaps we can have the T cells be on. We can make sure that any new T cells that try to respond to that tumor can't get turned off. And we can sort of heat up that immune response. Not only are there situations where people will use this with CTLA4 as a negative regulator, some people will instead think about other negative regulators like PD-1 and use an antibody against PD-1 to block PD-1. It's the same idea. We're basically trying to block negative signals so that T cells can't get turned off and they stay turned on. The idea is basically trying to stop this evasion of the immune response. The most famous of these is an antibody called pembrolizumab, or Pembro, sometimes also known as Keytruda, is one of the trade names. And some of you all may have heard of something called checkpoint inhibition. This is checkpoint inhibition. Checkpoint inhibition is just using something to block these negative signals, to block CTLA4 or PD-1, so that basically you can try to make sure that you don't get those negative signals. You're trying to, oops, wrong direction. You're trying to stop the evasion, keep the immune system going. You're trying to sort of keep things immunogenic and thwart the tumor in its ability to get around the immune response. There are lots of different ideas about regarding this idea of kind of like just taking the immune response to the tumor and turning it back on, or waking it back up, or heating it back up, whichever metaphor you want to use. Just because it's sort of interesting, I wanted to mention to you, this is a huge paper in the field. It came out during this semester. And what the investigators actually realized in this situation was they looked at some epidemiological studies and saw that when people were starting to get COVID vaccines, some of them were having less cancer, or they were having fewer problems with their cancers. And they actually found that when people started getting mRNA vaccines, that basically was sort of giving them a little bit of an inflammatory response. And that response and the way that their tumor had been evading the immune response, suddenly the immune system woke back up and was like, whoa, I got to deal with this tumor. It was basically like in vaccinating many of these individuals who were in very late stage tumor settings. In fact, the immune system seemed to take that cold tumor and get heated back up upon vaccination. This is a very, as you can see, new finding. This is one of the figures from this paper, though it's there's a lot that can be said, where they basically were able to see in these blue lines that these patients with various types of cancer survived much better than if they had been vaccinated, which is in the blue line, as opposed to those who had not been vaccinated. So SARS-CoV-2 doesn't do anything. COVID doesn't give you cancer, but it looks like actually having immune stimulation may help you with cancer therapies. And many of these patients were on things like checkpoint inhibition, and it seemed like their immune system just generally worked better having gotten a vaccine. This was one of the papers that was an option for the primary lit assignment. So if this is something that is of interest to you, this paper, as well as a link to a podcast episode where my colleagues were talking about it, I wasn't on that episode, are both in that assignment. And it is sort of a cool area of research. One other kind of big picture idea that some people have is sort of this idea of thinking about doing kind of a tumor vaccine or a cancer vaccine for different people. Remember, we talked last time about this sort of problem of, well, we could totally vaccinate you against tumors. We just don't have a time machine to know what tumor we need to vaccinate you against until after you already have it. And at that point, your immune system's already tired. Your immune system's already in this evasion phase. And so in this idea of cancer vaccine, basically, we will have a patient who has a tumor and we'll do some surgery to take out some of that tumor. So we'll excise some of the tumor. And we'll actually take that tumor and put it into a culture dish with some T cells also from that patient. We might then give those T cells IL-2. We would give those T cells other stimulation. And basically what we would be trying to do is wake the T cells back up in a dish. So the T cells have been silenced, maybe through cytokines, through other kinds of signaling in the patient's body, because the tumor is actually trying to silence the T cells. The T cells are exhausted. The T cells are not working anymore. So we take the T cells out of the patient. We don't know what the antigen is. It would be really hard to figure it out. So we just take some tumor and use the tumor to stimulate the T cells. We do this in a dish. We get the T cells all nice and revved up and ready to go. And then we put the T cells back in the person after we've basically expanded them in culture. And when we do that, we often see, so here you can see sort of a T cell transfer. And in fact, our patients will tend to do pretty well in having this happen. But of course, it would have to be person by person. You'd have to take each person's tumor, each person's T cells, make a personal vaccine or make personal expanded activated T cells for that person and then put them back into that person. You can't like do this in bulk and like sell it in bulk. You have to make it individually for each person because you'd have to be thinking about their tumor, their antigen, their MHC, all of that kind of stuff. David, I saw you had a question first.
So the T cells are taken from the tumor because those T cells that are near the tumor are already reacting. Sort of.
You're probably getting tumor-specific cells there. Here it's showing them coming from the tumor as well, but you might also take blood. You could do it either way. Yep.
How does this differ from a CAR T, or is this literally just the CAR T?
No, it's not a CAR T, but the CAR T is the next slide. So, so this is one way that we can do this. You can imagine that this is a little bit, you can imagine some of the drawbacks of this, that how personalized this would have to be. And when I talk about that personalization, you can imagine how expensive it would be per person to have to do this for each person individually and how much time it would take and things like that. So what some people might say is, okay, we have antibodies that we know work really well in the lab against certain tumor antigens. And some of them are even against these sort of conserved antigens that are frequently found on tumors, these tumor-associated antigens. That could be something overexpressed, like HER2 in certain types of breast cancers. That could also be something like CD20 that's on these leukemias and lymphomas that I told you about. And so sometimes we say, well, yeah, we got these antibodies and those are cool, but what if we could actually design a T cell in the lab? What if we could actually do our own sort of VDJ recombination and do our own biochemistry and make a T cell receptor in the lab that we want? And so instead of treating with an antibody against CD20 or an antibody against whatever the tumor antigen is, what if I could design a T cell myself that could bind that antigen in the lab, design my own T cell receptor, make a T cell with my personally designed T cell receptor, and have that work? Because T cells are, pretty good at killing. Antibodies are just like a protein. T cell actually is like, it's going to have more oomph behind it. And so that is the idea that some people did. And when they started with this, they started thinking about CD20 as their antigen. So they were thinking first about certain types of leukemias and lymphomas that have this antigen CD20 on them as B cells. And they said, they said, we know there's this antibody. It's a great antibody, rituximab. We know it's heavy in light chains. We know it's VD and J to make the heavy and light chains. We know all about rituximab. But it's an antibody. And, Dr. Barker loves T cells. So instead, what they did is they would take a patient and they would take cells from the patient. Basically, they would take T cells from the patient. So here's my patient. We take T cells. And then in the lab, they genetically modify those T cells to give them this designer receptor that they designed. So they have actually designed a receptor, a T cell receptor against the antigen, against the tumor antigen. So now they can get T cells acting against the tumor antigen. So they're basically going to genetically modify these T cells. As you will see, they're going to then potentially put those T cells back into these genetically modified T cells with this designer receptor back into the patient and have this work to kill the cells in the patient. Here you can actually see a picture of the first person who ever had this therapy done. So this is actually the first ever patient, Emily Whitehead, who had this done. So one thing that is really cool that don't have time here to really talk a lot about is the way that they actually genetically modify the T cells to put in the receptor of interest. They actually use a virus and a bunch of parts of viruses. And so that part, again, I could geek out about, but I'm not going to geek out about here. But we can also think about how exactly they designed this receptor. Because remember, when we talked about T cells, we talked about, well, you need signal 1 and you need signal 2, and sometimes you need other signals, and there's a lot going on with T cells. And we need to make these T cells be active. And so what they actually have done is they take the heavy and light chain, the DNA encoding the heavy and light chain of the antibody, because they know that binds to the antigen. They don't even care about MHC here. They basically just make this T cell that has a receptor that can bind directly to the antigen because it has the heavy and light chain parts from that rituximab antibody. And then it has parts of CD8, parts of CD3, parts of all of the other molecules that we need to signal a T cell, all spliced together. So this one receptor can both recognize the antigen and do all the signaling because it has pieces of all of the important signaling proteins. So this protein that they have made in the lab and are genetically modifying into these T cells does not have a real T cell receptor. I mean, it probably does have a T cell receptor, but no one cares. It has this weird fake receptor. It's not really a T cell receptor, though it has some things like a T cell receptor, and it's on a T cell. It's not really a B cell receptor. It's really a Frankenstein receptor that has a whole bunch of pieces of a whole bunch of different proteins, which means it is chimeric. It has pieces from different places. And it's not a B cell receptor. It's not a T cell receptor. It's this weird Franken thing. It's an antigen receptor. It just binds to an antigen. And so we call these T cells that have been genetically modified with this new designer receptor CAR T cells, for chimeric antigen receptor. And so the idea is, instead of using an antibody, let's actually design a receptor based on that antibody, put the T cells into the patient, and then kill the tumor. And so here you can see, here is this CAR T cell. Yeah, it's got a T cell receptor. No one cares about it in this case. It also has a bunch of copies of this chimeric antigen receptor that are going to bind to some antigen. Here it's showing it as CD19 instead of CD20. I don't know why. I don't care. It's some antigen on the surface of our cell, and so that's going to allow this CAR T cell to kill the tumor. And again, you can see sort of the Frankenstein-ness. There's some CD28. There's a whole bunch of things that are going to allow this T cell to get activated through this receptor. Yep, Jazz.
There's so many advantages to CAR T, but why would you, I know you're not immunologist MD, but immunologist PhD, why would you use a TIL instead of a CAR T?
Because a TIL is, why would you use T cells from the patient that you don't genetically modify? So Jazz's question is, why would you just take the patient's T cells, culture them in the lab with tumor antigen, instead of taking the patient's T cells, genetically modifying them, and putting them back in the patient? Couple reasons. One, you have to know the antigen, and you have to have an antigen receptor to genetically modify. If you don't know what the antigen is, you can still just culture and know that it's in the tumor and not have to worry about it. So if you can't design a CAR or a receptor, or if you can't, you don't know what the antigen is, then you can't do this. This is also, you also do have, I said may, but do, there are some downsides to doing a genetic modification like this. And so in some cases, sometimes the genetically modified T cells have had, the CAR T cell have had some issues. Every so often the CAR T cell becomes cancer itself because of the genetic modification. So you're sort of running that particular risk as well. The other thing that I will say, and again, here you can see sort of that CAR T cell killing the tumor cell of interest. Here is Emily Whitehead. This is actually from 2023 when she started her freshman year of college. So she's basically about your age, and this is some information about her, and she was the first person to have received this. CAR T cell therapy has been FDA approved, so it's not just a thing for Emily Whitehead now, it's something that can happen. It was the first type of gene therapy approved in the United States. When they approved it as something called Kymriah, when they approved Kymriah, it basically had it cost $1 million. And so the other reason is this is so expensive. And so doing a version that doesn't actually include the genetic modification could be cheaper, though not a ton cheaper, to be perfectly honest. They're all still out of reach for many people, but this is also like stupid expensive. So that's the other reason, is the other one might not be quite as expensive. CAR T cells have worked really well in certain situations. They have also had issues. Emily Whitehead had a bunch of problems when she got her CAR T cell therapy. Her CAR T cells were responding to CD20, which is on all B cells, as I told you, with rituximab. So she actually had a T cell response that killed all her B cells and was immunodeficient for quite a while. She also had a massive cytokine storm and almost died immediately afterwards until they gave her a lot of cytokine blockade. So this is not something that like is easily doable or easily worked out to be done in like mass numbers of people, though it's still being worked on. To be perfectly honest with you, CAR T cells are sort of going out of fashion right now. People are spending way more time with the antibody type of drugs than with CAR T cells at this point, like way more. I know people who work on CAR T cells, but if you think about the pharma industry as a whole, CAR T cells are like definitely on the outs. What are you gonna say?
Is there, I guess, a hybrid between TILs and like CAR Ts? If you take the T cells from the tumor and then chimerically transform them?
I mean, you could, but why?
I guess for hybrid special.
No, because like you're at this point you don't care what the specificity of the T cell was because you're putting in a different receptor. You don't
That's good.
okay. You don't care about the T cell receptor. So why do all the work of getting TILs when you can just draw some blood?
Sure.
Like there's no benefit to pulling them out of the tumor at that point. So there is another cool therapy that I think is amazing. That I just want to mention that involves a lot of aspects of immunology in terms of cancer therapy, and it's called oncolytic virotherapy. So what people have also realized is, again, our problem here is specificity. We have this problem of we've got this treatment where we're delivering chemo drugs or whatever very broadly to all the different cells of the body. We need a way to deliver things to only certain cells of the body. Something else we know that involves a lot of specificity is viruses. Viruses only infect certain kinds of cells. And so if your patient has a brain cancer, why not give them a virus that infects brain cells and maybe kills specifically their tumor cells? People have figured out ways to actually genetically modify viruses so they will only reproduce in tumor cells. And so the link that's here is a link to a 60-minute story about one of the there are many oncolytic virotherapy trials where people with incurable, inoperable brain cancer were actually treated with a modified poliovirus. They were infected with this virus that could only replicate in the tumor cell, and their tumors were basically cured, with the idea being that the virus was actually infecting the tumor and lysing that tumor. And so in addition, thinking about ways that we can use viral infection to actually lead to this therapy can be really useful. One other thing people have realized, I will be honest with you, I don't have a great answer of why I have, well, I guess I sort of know why I have the slide too about oncolytic virotherapy. The original answer of how oncolytic virotherapy worked is that you infect with the virus, and the virus is killing the tumor cells. But what we also are realizing is if you infect tumor cells with a virus, that also just heats up the immune system there. That also just wakes up the immune response. So you might not even need the tumor to actually kill the cells. It might just be enough to wake up the the virus to actually kill the tumor cells. It might just be enough to suddenly make the tumor immunogenic again, to make the tumor a hot tumor again instead of being a cold tumor. And so there are a bunch of places where now we're like, oh, actually, maybe it's just the inflammation of that virus. It's not the virus doing the lysis in and of itself. And so again, this is sort of a really cool area of research, but one where where you would also have to have taken virology for me to go into it in further detail. All right, Emily.
So I know you talked about antibodies being like a way to heat up the immune system, but also like the CTLA-4 and PD-1 blockers. Could you technically just like inject cytokines in the general area of the tumor?
So first of all, the PD-L1 blockers and the CTLA-4 blockers are antibodies. Okay. So they're antibodies that are blocking them. So when I say, in fact, when we're heating up the tumor, it's kind of because we're blocking the turning stuff off.
Okay.
So Emily wants to know if you could just inject some cytokine into the tumor area. In general, the answer is no. If you think about everything we talked about this semester with regard to cytokines and a lot of cytokine responses, in general, cytokines have evolved to be super tightly regulated in terms of time and location of presence. And we do not have a way that we can inject them as locally and as controlled as that. So there are a few cases where we treat people with cytokines for things, but they're on the rare side. And a lot of times when we have tried to treat people with straight cytokines, like now we sometimes do some modifications, bad stuff happens. Because in general, we can't do the treatment and get in the Goldilocks zone. We only can do the treatment and give you way too much. Sometimes I show Dallas Buyers Club in my DSEM, which is about HIV, and the main character is trying to get a bunch of HIV treatments. At one part of Dallas Buyers Club, he actually gets interferon from a source and injects himself with interferon, kind of for the same idea of trying to inject himself with interferon to get rid of the virus, and then promptly has a heart attack. So in general, we are not at a place where we can just deliver the right amount of cytokine at the right time, at the right level, to have it be in any way beneficial. We are not that good at Goldilocks. So I also want to talk a little bit about treatment for some of the other diseases that we've talked about, specifically thinking about autoimmunity for a second. So we've already talked about some autoimmunity treatments, specifically using things like steroids or using things like cyclosporin A. And they're all great. But if you notice with steroids, we're sort of getting this super broad immunosuppression. We're suppressing kind of all sorts of aspects of the immune system in a whole bunch of ways, with prednisone or something like that. When we use cyclosporin, we're doing a little bit better. Instead of suppressing every single part of the immune system, now we're only going to suppress T cells. That's a little better. And remember, cyclosporin is actually acting on our NFAT signaling pathway, so we don't get T cell activation because we don't get NFAT. So we've gotten somewhat better, but again, we would really like to be more specific. We'd really like to not just treat someone with cyclosporin and impact all of their T cells, but what we'd love to do is say, let's turn off just your autoimmune T cells and leave the rest of your T cells okay. Because right now, if you're on cyclosporin, if you have an autoimmune disease, we treat you with cyclosporin for the rest of your life. That means you're immunosuppressed for the rest of your life. One of the medications that comes up in a later slide, my dad has been on for years. And at one point, actually, relatively recently, like during COVID, he looked at me, he's like, wait, am I immunosuppressed? And I was like, yes, Dad. He's like, wait, so everyone else's vaccine protects me in herd immunity? And I was like, yes, Dad. And he's like, well, I'm going to have to tell them to get it then. And I'm like, good. But the point that I want you to see here is that, again, our problem is sort of breadth in therapy. And we got a little bit narrower, but we want more specificity. Oh my gosh, we have this whole system that's about specificity. And so for most autoimmune disease treatments now are what are known as biologics. But honestly, the biologics used for autoimmune disease treatment are just antibodies that may have been modified in a variety of different ways. Also, this semester, this amazing review of use of antibodies in therapy and in clinical settings was published. So I'm going to show you a couple things from it, but if you want to know about how the pharma companies use antibodies, this has all of it in there. And so just to point it out, these are some figures from this paper. This is actually showing you the increase in number of drug approvals for antibody drugs. So you can see that they kind of started to be a thing in the sort of early mid-90s. And you can see that, there were a couple approved in the 90s, and now we're at like 250. And so these drugs are just sort of taking over the pharma industry and are becoming the key kind of drug. The top is kind of telling us about the types of technologies that are used, which I don't want to get super into. The bottom one, however, tells us what applications they're used for. And so you can see in this light blue, A bunch of them are used for cancers, like we just talked about. And then in yellow, a lot of them are also used for autoimmunity, which we're talking about now. But they can also be used for transplantation or other things, and also for a bunch of non-immune related things. So other people are coming in and stealing our tools and using them in a bunch of different types of drug therapies. Yep, Nadia. So basically what you are getting in these cases is you are getting, you are taking injections of your biologic for the rest of your life. Okay. So basically you are, if you're doing this, if you have an autoimmune disease, you have these T cells that are attacking your body, for example, you are going to, you're going to try to suppress them. Yeah. If you ever stop suppressing them, they'd come back and hurt you again. So you basically are going to inject the antibody to try to keep them turned off for the rest of your life. Yes, I'm doing it like this because when my dad does it, he injects himself into the quad. Also, I like watch him do it, and having been like trained in doing different types of injections, I like freak out because I'm just like, no, that is not how you do any of this. Yes.
So if you're taking cyclosporin A chronically, does that mean that you could be more predisposed to cancer?
Maybe you could be more predisposed to cancer. I don't know that we've seen chronic cyclosporin A for enough years to like have really seen that pick up, and we mostly worry about infectious disease situations, but potentially. Yep. Did you have a question, or did I just see things? Okay, I just saw things. That's fine. Here you can kind of see, this is actually just showing you all the different ways they've made just rituximab into different kinds of treatments. So they've made it into a T cell receptor in CAR T cells. They've put chemicals on it. They've put all sorts of different kinds of other things they want to deliver to the cell. They've designed it in all sorts of cool different ways. And so because of knowledge of antibody structure, there can be all sorts of cool modifications of these where you can kind of hit your cell of interest with whatever you want, as long as you know what the antigen is. And so in general with the biologics for autoimmune diseases, what we're trying to do is we're trying to narrow down that therapy. And so sometimes we use things like an anti-CD20, and we're getting rid of activated B cells. So at least that's better than all B cells. Like at least maybe it's going to be a little narrower. And that might be, and rituximab is in fact used for rheumatoid arthritis. We might use an antibody against CD4 to sort of get rid of CD4 T cells. At least then you have CD8s. So again, we're narrower, but it's still not like we're only getting the bad T cells. The bigger thing that we often see happen might be something like this, where we might use an antibody. So another kind of molecule that looks like CTLA-4. Remember, CTLA-4 blocks B7. So this one is not trying to get to sterically inhibit CTLA-4 from binding stuff. This is actually using what we know about the structure of CTLA-4 to hide B7 and to make sure that we sort of don't turn on T cells. We're basically trying to make sure that more T cells can't get turned on and inhibit T cell activation. So it's sort of the opposite side of the interaction here. But we can basically say, your T cells are doing too much. We're gonna try to stop any more of them from getting activated. We're gonna try to stop T cell activation. And this is used, you can see, in a number of different sort of disease states. And you can see this exact thing happening here in this situation. You can see that basically we're blocking B7. We can't ever get signal 2, so we can't ever activate any more T cells. And so now we're basically preventing T cell activation. We're saying like, some of your T cells are bad. We're going to try to like tone this down. But at least, you know, there's some other co-stim molecules, so it's maybe not as complete, not all T cells, it's just some T cells, because there might be other co-stims. But in fact, the biggest place where we see a lot going on with any of these is the opposite of where Emily was thinking. So in fact, what we often will do is we will use antibodies to block cytokines. So we might know that your disease is caused by Th1 cells. And so instead of blocking all T cells, let's just block one of the Th1 cytokines. So that's going to get us narrower. It's still not totally specific. You still are immunocompromised because you can't make Th1 responses to microbes where you need a Th1 response. But it's way narrower than before where we were hitting everybody. And so people use different antibodies that will block different cytokines or use mimics that block receptors. And so we see cytokine blockade as actually far more common as a thing here. And so, you may have heard the two particularly big ones. Tocilizumab was used during COVID to block IL-6 when people had too much IL-6 and too much inflammation. You may have heard of Enbrel or I don't know why I can't think of the name. There's another one that's really famous, whose name I cannot think of right now, that are used in rheumatoid arthritis. Yeah?
Methotrexate?
No, methotrexate is more like steroid. Oh my gosh. Huh?
Skyrizi?
No, Skyrizi is new. It's IL-17 blockade.
Make something up.
No, so there's two for TNF blockade that are like the big ones. One of them, it's Hannercept. One of them is called Enbrel, and the other one I feel like starts with an R, but I cannot remember the name of it to save my life. But like the idea of just turning off TNF. Remicade. The idea of just turning off TMF and sort of trying to block some T cells, or at least the pathologic effect of the T cell, is sort of where people are with a lot of this type of stuff now. There are so many diseases that are now treated with cytokine blockade. There's now an IL-17 blockade. There's an IL-23 blockade. There's one, I can't remember what cytokine it's blocking. I'm thinking IL-9, but I'm not sure if that's correct. That they talk about, that they suddenly are like, it's going to help your asthma. And they go on and on about how it's helping your eosinophils. And like,
I saw a commercial.
yeah, and it shows eosinophils in the middle of the commercial. Yeah, that's that one. And so this is like the place where most of the people are going, is trying to sort of do this more narrowly by blocking certain cytokines. And we keep increasing the number of cytokines that are sort of options. Many of the things that we've been talking about, of course, would work for treating some of our hypersensitivity responses that are types 2, 3, and 4. But if you recall, a type 1 hypersensitivity response is unique because it is IgE and mast cell mediated. One thing I forgot to say on the previous slide, there actually was also, so I get table of contents of different journals come to my inbox, my email inbox. And yesterday was like a big day of like journals. I got so many journals in my inbox and new papers yesterday. There was also a really cool review about people trying to use CAR T cells for autoimmune types of things, where somehow, and the idea there is the antigen is actually the autoreactive T cell receptor. And so somehow you have to have figured out what the autoreactive T cell receptor is and try to kill just the autoreactive cells. It's really hard, but there was a big review on some of the progress in that. There are definitely some people working on that. Who knows? But anyway, that was the thing I forgot to mention before. So oftentimes when we are seeing allergy treatments, because when you're at the point of needing an allergy treatment, you already have IgE, you already have class switch, you already have a lot of response. Most of what we're going to be able to do is just block the mast cells and block their effector function. That might happen with antihistamine, that might happen with an EpiPen. All of these things are really kind of blocking the things downstream of the mast cell. And that's probably the biggest place where we're going to see things work for a lot of types of allergies. There are some ideas of treating people with antibodies against IgE and basically trying to remove your IgE. And so if you already have some kind of bad IgE in your body, can we give you an antibody against IgE to just clear it all out? And so could we give you an anti-IgE? This is sort of a thing that's in experimental use. The other big thing that is out there for treatment of different allergies is basically a situation where we try to induce tolerance. So the problem here is you have too much immune response and we would like you to have tolerance. And there are sort of two different ways, really they're kind of two different angles of the same way, that people can try to induce tolerance. One of them is to have you eat the antigen. And so sometimes we actually have you eat the antigen because we know that in the GI tract, if you encounter antigens by the GI tract, like in food, you tend to make more of a Treg biased response. It's actually shown better on this later slide. And so what we might do is we might say, okay, we might start, we know that in fact the immune responses that happen through the GI tract tend to be tolerance and tend to be sort of more IgA, Treg responses. And so when we feed you antigen, so if you have a really bad allergy to something, sometimes what we do is we actually try to feed you small amounts of the antigen to see if we can push you into making Tregs. The other thing that we try to do is sometimes we will actually try to basically keep giving you the antigen like in your arm or something and desensitize you to it. So the idea is basically that we are trying to either do clonal deviation or we're trying to exhaust the cells. We're trying to deviate them to Tregs, deviate them to anything that's not causing you problems, deviate them to anything that's not Th2, or deviate them specifically to Tregs, or even to potentially exhaust that cell. And so basically if we keep giving you the antigen, we might desensitize your response to it. If anybody gets allergy shots, that's what you're doing. You're basically getting low amounts of antigen and trying to exhaust your T cells, trying to exhaust out that response. Some of these, when you look at them, you're like, that sounds terrible, because it basically says you're going to go find the thing I'm allergic to and then give me a bunch of it. And these aren't really effective in like making allergies go away completely, but especially with some of the like oral sensitization methods, there are some cases where they've been pretty good in terms of like a little kid who's so peanut allergic that they can't be in a room where somebody opens peanuts. Like they would die if somebody in their kindergarten class opens peanuts. They can actually get to the point where their allergy is less severe. And then they can sort of do daily life. They're still not eating peanut butter, but they can actually kind of make it through without being super worried about environmental levels. And so most of these cases, we can see some good reduction of the severity of the response, but we can't make the response go away entirely. Yep, Miles.
So would this not work for you because your blood wasn't a problem?
I don't know what would work for me at this point. I think that what the biggest issue is to do this, you need to know what the antigen is. And since I came back negative for all antigens, we don't know what my antigen is. And so in theory, part of the reason I got tested was because I wanted to know what the antigen is, and then I might have tried to do some of this. But so who knows? Like, I think in cases like what I have, they don't really have enough knowledge of like what to do. But it probably wouldn't help me. The trees know what they did. So the other place where we use our knowledge of immunology is in thinking about vaccines. And the idea of vaccination has a really long history. And it was originally thought about by a whole bunch of people who did not have the Bio 250 prereq for this class, but they still figured all of it out. In the Peloponnesian War, so this is one of the early reports that I can read, though there are similar reports in Arabic and Chinese texts, and actually some Sanskrit texts like really far back. People were noticing that if you survived, those who had recovered from disease never had to fear again. The idea was if you could live through infection, if you could be exposed and live through it, you would somehow get this memory response that would help you out. This was something that people had been doing in many parts of the world for a long period of time. They were actually deliberately giving themselves versions of viruses in order to try to give themselves immunity. Lady Wortley Montagu was the British ambassador to Turkey's wife. She saw this and wrote to people, and it was basically like, oh yeah, it turns out that if you just give people like a little bit of smallpox, like if you basically like scrape a scar off of a smallpox patient and then like grinded it, like cut your skin open and put it in, so you just give them a little bit of kind of dead smallpox because it came out of a scar, you're protected and don't get smallpox. I mean, unless you die from the whole scar thing, but some people did, but everybody else was doing pretty well. They also realized that you could actually inhale the scabs, so inhaling people's smallpox scabs. People used to actually mail smallpox scabs in the mail. Historical societies today still from time to time open up envelopes and find them full of smallpox scabs. Because it was a really scary disease, and this was like the only way you could protect yourself, was sort of try to induce this memory response. And so the idea was previous infection, as we've talked about, pre-existing immunity can protect you with a memory response. The bad news about all of that, of course, is you have to go through the first infection. And you actually saw this in the Bureau paper as well. You saw situations where infection was really helpful for the mice in the 75% who lived. And so if you don't die, this is great. And so the question is there a way we can take advantage of this knowledge to give you the same effect without having a risk of dying the first time? And so a bunch of people did some experiments, and basically Edward Jenner is often credited as being one of the first. He kind of realized that, well, if you actually take cowpox, it's a virus that's very closely related to smallpox virus. It doesn't cause disease in humans. And if you give somebody cowpox, that actually gives them that same protective immunity, and they're protected against smallpox. He did this by actually giving a small boy cowpox and then giving him smallpox and seeing that he was fine. They called it vaccination because this came from a cow, and Latin for cow is vacca. So it was vaccination. We actually now know this whole story is a lie, and the virus he used was actually horsepox, and so technically it would be like equination or something, but we still use vaccination. And so this is sort of the general idea behind all of this. And so again, the basic idea, and yes, I'm sort of skipping ahead a little bit because I have so much stuff that I could have had in here, is that we know that, you know, your first infection allows you to make a secondary response and does this memory response. And so the goal is to give you some preparation of the vaccine to induce a primary response so that if you really ever are infected, you have that secondary response in a safe way that didn't give you a risk of dying the first time. And this sort of piece of it, of thinking about vaccines, kind of makes a lot of sense related to a lot of things we've talked about this semester. You know, if you want to know what's actually happening during all of these things, go back and do the whole semester. So I don't want to go through all of that. You can kind of understand the effect that we're having with this preparation. I don't know that I'll have a ton of time to tell you what the preparation is. One thing that I will also tell you is that it induces both an innate and an adaptive response. Sometimes when you get a vaccine, you get a fever. Sometimes you feel a little crappy, right? It's not because it gave you the disease, it's because you're making an innate immune response. We actually don't know how to give you a vaccine that doesn't make an innate immune response, and we probably wouldn't want to. There are some studies out there that were done during COVID vaccine times that having worse side effects were correlated with making a better response. So they actually looked at a bunch of nurses who got the vaccine really early, and the ones who had the worst side effects made more antibodies. So that innate immune response that makes you feel crappy might also be making your vaccine work really well. And so whenever one of my friends says to me, oh, my vaccine made me feel so bad, I'm like, I'm glad your immune system is working as it should. And as I usually joke here, no one has punched me yet when I said that. So vaccines are going to give us all of these really critical individual level effects. They also lead to a number of different effects at the population level. So we don't just think about the sort of general immunology pieces here, we also think about what's going on at the population level. Because basically, it's possible that when you are vaccinated, you might still get infected. And you can see that on that previous slide as well. You can see that in the memory response, we still have a little bit of virus replication. It's just a really little bit, not enough to make you sick. It's also probably not enough to spread, which means that you getting vaccinated both helped you not get sick, but it also blocks spread in the population. So we're now not going to see transmission among members of the population. We're basically breaking the cycle, and our population is protected. Having sort of a general level, and so what percent of your population has immunity is also really important. For each different microbe that's out there, the probability of spread basically drops off when you get to a certain threshold. That threshold number is different for different pathogens, has a lot to do with the biology of those different pathogens. There also are things related to different populations, so you always see kind of a range here. And so you might look at this and be like, oh, that's awesome. So what you're telling me is I actually don't need to get vaccinated. I just need to make all my friends get vaccinated, and then I'm good. You can actually see, here's an example of this. Here's the one infected person goes into a community where everyone's susceptible. Spread happens throughout. If a bunch of people are immune, we block spread. And so you might be like, sweet, I just have to make sure all my friends are vaccinated. The thing, of course, that we have to realize is there are some people who are immunocompromised, like my dad, who need our help in getting protected. And in fact, as we're treating more and more people with cyclosporin, and we're treating more and more people with these cytokine blockades and things like that, the number of immunocompromised people in the world is increasing. There are also people who can't get immunized for other reasons. There are people who have cancer or who have other immunodeficiencies. And no vaccine is going to be 100% effective. And so we all kind of have to do our part in getting vaccinated in order to really help our population's level of vaccination. I'm not going to have time to show you the videos that kind of talk through or show this pattern in picture form, but if you actually want to watch how percent of people vaccinated changes over time, you can see that here. This phenomenon is usually called herd immunity, though during COVID people got mad at that and they didn't like being called cows or something. So immunologists tried to start changing the term to community immunity, which is really hard to say. So sometimes you'll hear it as community immunity, but it was originally herd immunity. This is one important way that knowing about immunology impacts your life, and there are many other things one could think about with immunology impacting their life. But in my last 1 minute, I'm skipping to the end of the slides. Hey, if I can figure out how. Why is it when I don't want the little menu bar? I get it. But I want to tell you two other things that are important about immunology in your daily life. One, many immunologists like to say things about this. In immunology, we talk a lot about why it's important that we have extreme diversity through VDJ recombination and the broad diversity of the adaptive immune response. We say that's the only reason adaptive immunity works is from extreme diversity. So you should learn about the importance of diversity and being part of a diverse team from the immune system. Also, yesterday in my emails, when I was getting emails about, when I was looking at different papers, this paper was published yesterday called Immunology as a Guide to Human Relationships. And I printed out copies of this kind of silly philosophical paper in case you want to look at it. But it reminds us of things like we should calibrate how much we recognize things and make sure that we have confirmation that something is a threat. We should reduce things in the background that stress us out. We should try to regulate instead of kill. We should try to have memory and revise and learn from our experiences. And we should make sure we have clear communication. And so hopefully throughout this semester, you learned a lot of things that are going to just help you in everyday life, even if you don't think about immunology. I laughed so hard when I saw this yesterday that I was like, I have to. So anyway, this has been a really fun semester. It's been really cool to have you guys and how engaged and excited about things you have been. So thank you guys so much. I'll see you for the review session tomorrow on Zoom at noon, and then the exam here on Monday at 12:30. And I have this important thing about immunology and your human relationships here if you would like it. So thank you.